Okay, so we're back to these determinants of oncogenesis. So these are all those regulatory genes I mentioned that are frequently mutated in cancer. So initiation, or sorry, neoplastic transformation starts with a mutation. It's usually in one of these four classes of genes. Mutations in these genes cause cancer. So very briefly, we have proto-oncogenes. These are normal gene, all of these are normal host genes that when mutated cause cancer. Does that make sense? So a proto-oncogene is a normal host gene. Its goal is to promote cell proliferation when you want cell proliferation. When it's mutated, it'll be always on, which means always proliferate. That's cancer. Tumor suppressor genes are kind of the flip side of proto-oncogenes. These are genes that usually inhibit cell proliferation. So they're like the brakes on replication. If they're mutated, the brakes are off and we can proliferate with no checkpoints. That's cancer. Apoptosis regulatory genes. So remember, apoptosis is one of the mechanisms we have to control initiated cells and neoplastic cells, right? Cells that are abnormal, we can have them undergo apoptosis to get rid of them. So these regulatory genes usually inhibit apoptosis. We talked about briefly, I think, during apoptosis, you have a level of pro and anti apoptotic factors in your cell, and they're always kind of going like this. So if we always activate our inhibitory apoptosis genes, we get no apoptosis. That's cancer. And then DNA repair genes. So these are the enzymes that repair errors in DNA. If they're non-functional, cancer. Make sense? Here's the cell cycle. Everybody know it and love it? Where do most normal adult non-stem cells spend most of their time? They're riding around the cell. G0. How'd you know that? So you guys are smart. Yeah, so most normal, healthy adult cells at any given time are not dividing. They are not riding the cell cycle. They're in cell cycle arrest. Where do tumor cells spend their time? Do you know you haven't told us?
Say S.
Just on the cell cycle. So a lot of people, I was trying to get you to say mitosis because we see mitotic figures, we count mitotic figures. We see more mitotic figures in neoplastic cells because they're always on the cycle. We don't see them in adult normal healthy cells because they're usually in cell cycle arrest. Right? Does that make sense? So cell cycle checkpoints. We all have this in undergrad. You get it phys, immunology, anything? No, just undergrad. Okay. Can we do a show of hands for my education? Who had this in undergrad and feels pretty comfortable with cell cycle checkpoints? Who did not? No mockery. I seriously kind of, yeah, okay, cool. All right, so we'll go into a little bit. The cell cycle checkpoints are basically these regulatory steps in the cell cycle. Basically what we're doing is we're checking the cell for DNA damage, any other issues that would make us maybe not want to divide because we could become cancer if we divide. We don't want to let an initiated cell divide if we can help it, right? So these checkpoints are all the control mechanisms where we can stop, check the cell, be like, hey, are you okay? You got any damage over there? Any mutations? Feeling particularly neoplastic today? No? Okay, you can go ahead and divide. So if we do find one of those things, we stop the cell cycle, we arrest it, and that prevents replication of cells that are mutated. So if you're a damaged cell and you are caught here holding dirty DNA at one of the cell checkpoints, you have two options. You can undergo repair, you can get right with the host, and then you can resume the cell cycle and go about your business. If you can't repair, you're undergoing apoptosis. So these are the control mechanisms. So the molecules that control these checkpoints are called cyclins and cyclin-dependent kinases. Cyclins, like the cycle, they actually rise and fall in a cycle. So you have to reach a certain level of cyclins to progress to the next stage, like this. So these levels of cyclins rise and fall. In order to go from G1 into S, you have to have this much G1S cyclin present. And then as you go through the cell cycle, it goes down and these others rise, and that tells you you're okay to enter this phase. So, back to the meat. Proto-oncogenes, again, a proto-oncogene is a normal host gene that promotes cellular proliferation. It's normal. If it's mutated, it can become oncogenic. A mutated proto-oncogene, we call it oncogene. It's not proto anymore. Proto means like a baby, a gene, onco being cancer. Now it's a cancer gene. It's mutated. A lot of these are growth factors or growth factor receptors. Some of them are cell cycle regulators. So cell cycle regulators like cyclin-dependent kinases. So if this is mutated and your cyclin-dependent kinases are always on, that means they're always phosphorylating cyclins. And these levels are always up. And so the cell is just like boom, boom, boom. I can fly through every checkpoint with no stops. Okay, so importantly, mutation in proto-oncogenes to make them oncogenes. These are dominant mutations. So you only need one hit for an effect. Does that make sense? Versus a recessive, you need 2 hits. Also in the genetics module, check it out. It's great. So a mutation is dominant. You only need 1 mutation, and we need a gain of function. So proto-oncogene promotes cell proliferation. We get a loss of function, that will not lead to cancer. Right? We have to have a gain of function. So this needs to be always on and doing whatever it's doing. So we're going to get an overexpression of these things, growth factors. We're going to be producing more growth factors. We're going to be making more receptors for the growth factors. Anything that increases proliferation. Questions? Here's an example. Anybody heard of RAS before today? Couple, nice. So RAS, I'm gonna skip before and show you how important RAS is before I talk about it. So don't learn to memorize this, please, ever. These are RAS mutations in human cancers. Look at these numbers. Pancreatic, appendix, pancreatic, bowel, colorectal, lung cell, all these. High frequency of RAS mutations in human cancers. So like I said, these are called driver mutations because these are the most common mutations in cancers, is in these class of genes. So RAS is probably one of the biggest ones that we think about. RAS is a proto-oncogene, so it's normal. Its job is to transmit growth signals from the growth factor receptor to the nucleus. So don't worry about all these molecules in here in this cartoon, but you have receptor. RAS is the middleman in signaling, transmitting these growth factor signals to the nucleus of the rest of the cell to activate all the stuff, okay? So if RAS is mutated with a gain of function, we don't have any need for these growth signals or receptors. We can just ignore them and just RAS away, right? I can just proliferate. I don't need the body's growth signals anymore. I am now autonomous growth. Importantly for us, this is also a potential therapeutic target, right? Because this is a middleman in this pathway. If we can knock this guy out, we can maybe save the cell. So lots of potential RAS small molecule inhibitors therapies under investigation in many different cancers. HER2, who's heard of HER2? Much more people than RAS usually, because this one is popular. So this is another, HER2 is a growth factor receptor in the EGF family. Here it is right here. HER2 is one that is commonly mutated or amplified in human tumors, especially breast, but also lung. So if we get a gain of function in this growth factor receptor, signaling will be always on. We don't need the growth factor to bind anymore. The receptor just takes over. Oh, look who's here. Who's that right there? RAS in the HER2 path, double whammy. So these guys are super critically important in many cancers.
This is the estrogen receptor.
This is an estrogen receptor. It's one of many. All right, so that's oncogenes, proto-oncogenes. The flip side of that, the opposite in many ways, are tumor suppressor genes. These are normal host genes that restrict or dampen proliferation of the cell. So a lot of these genes are involved in cell cycle control. P53 is a big one. Apoptosis of cells that are in cell cycle arrest, DNA repair. So there are things that like, whoa, stop replication and let's fix the damage and get back to the cell cycle. They're also called caretaker genes, which I think is really. So if mutated and activated, for a tumor suppressor gene to allow uncontrolled proliferation, it needs to have lost its function. Does that make sense? An oncogene needs to gain function. To promote neoplasia, a tumor suppressor gene needs to lose function, lose its caretaker function, its tumor suppressor function. Mutations in tumor suppressor genes are recessive. The oncogenes is dominant, so you only need one mutation. In a tumor suppressor gene, it's recessive, so you need 2 hits. This is called the 2-hit hypothesis. I've heard of that before. Means in a tumor suppressor gene, both alleles, both copies have to be mutated in order to cause cancer, because it's a recessive mutation. As long as you have one functional copy of your tumor suppressor gene, you can suppress tumors. Both copies are knocked out, tumors. P53 is the most popular tumor suppressor gene, often called the guardian of the genome. I'm not gonna ask who's heard of this because I hope it's everybody, and if it's not, I'll be sad and I don't wanna know. So P53 is guardian of the genome. It's a DNA binding protein. It can promote cell cycle arrest, cell cycle repair, or induce apoptosis. So this is a major control on damaged cells in the cell cycle and keeping them from replicating. Mutations in over half of human cancers, so this is even bigger than RAS mutations. Also mutated in many animal cancers. I was not able to find as clean of data of animal cancer. Very commonly mutated in many different types of cancers. So again, mutation in a tumor suppressor, you're going to get a loss of function. So you're going to get a loss of those checkpoints, loss of DNA repair mechanisms, loss of this apoptosis control in damaged cells. And then this is going to allow these mutations to become fixed in the genome, because it's going to allow these damaged cells to replicate. Anybody heard of BRCA? It's another one also in breast cancer. It's also been called the breast cancer susceptibility gene. This is another tumor suppressor where, similar to P53, it's responsible for DNA repair, genome stability, cell cycle progression. So again, it's a tumor suppressor. If you have a germline mutation, you have 1 hit, but you're okay. If you acquire a second mutation, you're not okay. So this is one of those genetic tests we do. How many mutated copies of BRCA do you have? If you have no mutated copies, you're okay. Both copies of that tumor suppressor gene are intact. You've got one in backup. Many people are born with 1 missing or mutated, so you only have 1 functional BRCA. So you are at risk of developing a mutation in your other copy, right? If you develop a mutation in 1 copy and you don't have another functional copy, that's a 2-hit. You have no functional tumor suppressor gene. Okay, couple summary slides on tumor suppressors and oncogenes. So oncogenes are like, proto-oncogenes are like the accelerator. Tumor suppressors are like the brake. In cancer, we are gain of function on the oncogenes. We're hitting the accelerator, and we've also deactivated the brake of tumor suppressor genes. So it's go all the time. Same information, different format for you to digest. Same information, different format plus a mean. Questions? I'll leave it at the beginning. Yeah.
Is there usually any kind of like sequencing for how tumors develop? Like, do they usually develop a mutation in a proto-oncogene, and then with the increased proliferation, have a mutation in the tumor suppressor genes, or is it?
That is a really
other
quick.
way around, or does it really not matter?
That's a great question. I don't know that it matters. And mutation in any of these genes is a driver mutation. So it could start with any one of these, but the more genomic instability you get as the tumor grows and mutates, that you'll get them in a bunch of these. So it always starts with one, and then there's kind of a cascade once your genome starts. But I don't know that any one is more implicated. Maybe tumor suppressor genes are slightly more implicated than the other ones. Good question. That's like a whole PhD in that question. Okay. Anybody want a PhD? I didn't either. Nobody wants a PhD. You can still do one even if you don't want one. It's good for you, like eating your vegetables. So our questions. Yeah,
If you have a pregnant female and a mutation starts to initiate in her, can it initiate and then proliferate in the fetus?
where in her is it initiating?
Let's say in the reproductive system.
Where in the reproductive system?
And
What cell?
placental.
What cell? Single cell. You gotta name a cell type for me before I tell you what happens to it. If it's a mutation in a neuron or a cardiomyocyte, they can't divide. I'm not worried. It's a mutation in a sperm or egg cell. Very worried. Also read about the epigenetics section in the genetics module. There are changes that could be passed from, so there are changes that if I smoke or have diabetes or there's a bunch of dietary things, I can pass that on not only to my child, but my child's child through epigenetic mechanisms. So epigenetics, little stickier, but for mutations, they really need to be in a germ cell. So they need to be in sperm or egg to affect the fetus. You could get mutations in the fetus germ cells, right? If mom consumes a mutagen that passes to the fetus, you could mutate the fetus's sperm or egg cells.